Welcome to General Biology. In today's video, we are going to compare mitosis and meiosis. Both of these types of cell division start off with parent cells with a normal number of homologous chromosomes. And in both cases, we are going to have to replicate the chromosomes before we can do cell division. So in both cases, during the S phase of interphase, we're going to replicate our chromosomes. And we're going to go through G2 just like normal, and then we're going to approach the cell division process, and then that's where things get a little different. So as the chromosomes condense during mitosis, the individual chromosomes stay separated from one another. In meiosis, the homologous chromosomes pair up. And so there's a cell signal that causes them to pair up. And when they pair up, they form tetrads and they start wrapping around each other. And an event called crossing over takes place. And some of the chromosome, the genes from your mom get swapped with genes from your dad. So your mom chromosome now has some dad genes, and your dad chromosome now has some mom genes. And this happens in every single pair of your chromosomes at multiple points on each chromosome. That doesn't happen in mitosis. In mitosis, the chromosomes stay independent. The mom genes and the dad genes are independent of one another. Next, we head into metaphase. In metaphase of mitosis, all of the chromosomes just line up in a nice row. And in metaphase of meiosis, they line up in their pairs still. So the homologous pairs are still connected to one another, and so they line up in their tetrads. And when we get ready to do anaphase, entire chromosomes get pulled in opposite directions during anaphase 1 of meiosis. In anaphase of mitosis, individual sister chromatids get pulled in opposite directions. Well, this process right here reduces the number of chromosomes in the cell in humans from 46 to 23. You can see here it reduced it from 4 to 2. Here, we don't reduce the number of chromosomes, we reduce the copies, but we had 4 chromosomes up here, we still have 4 chromosomes down here. Now, the second part of meiosis, there is no second part of mitosis. The second part of meiosis is then going to sort the sister chromatids into 4 separate gametes. So with meiosis, you end up with 4 cells, and mitosis, you end up with just 2 cells. You can see that the offspring cells, if we look at the chromosomes here and here, they're identical to each other. And if we compare them to the parent, they're also identical to the parent cell. So this process created clones identical to the parent cell. If you look at up here at the parent cell and you look down here, none of these are identical to the parent cell. And they only have half the genetic information that the parent cell has. So when we form gametes, we're going to create variety, but we're also going to create cells with only half the normal number of chromosomes. So when fertilization takes place, we get back to the normal number of chromosomes.